Hello everyone, today we will discuss about the metabolism of VLDL and LDL. VLDL is a lipoprotein that is synthesized in the liver cells. Its main function is to transport endogenous triacylglycerol or triglyceride from liver to peripheral tissues. That is the main function of VLDL, that is to transport endogenous triglyceride from liver to peripheral tissues. So let's see these steps in the metabolism. VLDL is synthesized inside the liver cells. Then ApoB100, it is synthesized by the polysomes on rough endoplasmic reticulum. Then triacylglycerol, it is synthesized in smooth endoplasmic reticulum. These endogenously formed triglyceride, ApoB100 and cholesterol, these together form a lipoprotein complex that is known as nascent VLDL. Nascent means newly secreted or newly synthesized VLDL. So, nascent VLDL, it will contain triacylglycerol, cholesterol, then ApoB100, that is known as nascent VLDL, newly secreted VLDL. Then this nascent VLDL is secreted into the circulation. It is secreted into the circulation. Once in circulation, it acquires ApoC and E. ApoC and ApoE from HDL. So, from HDL, it acquires ApoC and ApoE. And it is converted to mature VLDL. This nascent VLDL, on acquiring this ApoC and ApoE from HDL, it is converted to mature VLDL. That is, it will contain triacylglycerol, cholesterol, then ApoB100, then ApoC and ApoE. This is known as mature VLDL. Then the enzyme lipoprotein lipase. Lipoprotein lipase, this enzyme is activated by this ApoC. Here it is ApoC2. This ApoC2 activates this enzyme lipoprotein lipase. This enzyme catalyzes the hydrolysis of triglyceride. It catalyzes the hydrolysis of triacylglycerol to free fatty acid and cholesterol. It catalyzes the hydrolysis, lipoprotein lipase. This enzyme is activated by this ApoC2, lipoprotein lipase, and causes the hydrolysis of triglyceride to form free fatty acid and glycerol. Then most of the released free fatty acid, these are taken up by the tissue cells. Then glycerol is taken up by liver. So, as the lipoprotein lipase hydrolyzes the triacylglycerol, this triacylglycerol content in mature VLDL, it will decrease, and it is converted to this VLDL remnant. VLDL remnant, that is known as IDL, that is intermediate density lipoprotein. So, it contains less triglyceride than this mature VLDL. So, it is known as VLDL remnant or IDL, intermediate density lipoprotein. So, at this time, this ApoC2 and cholesterol are transferred back to this HDL. ApoC2, here it is VLDL, remnant VLDL or IDL or remnant VLDL. Then at this time, ApoC2 and cholesterol are transferred back to HDL. Then this cholesterol which is transferred back to HDL is transferred again back to this IDL as cholesterol esters. So there is esterification in place of triacyl glycerol from IDL to HDL. So from IDL to HDL triacyl glycerol, and from HDL to IDL there is cholesterol ester. This transfer of this cholesterol ester from this HDL to IDL, this is by an enzyme protein known as CETP. CETP, that is Cholesteryl Ester Transfer Protein. This is by the help of the enzyme protein known as CETP, Cholesteryl Ester Transfer Protein. Then this is IDL, that is remnant VLDL. Then in the liver there is receptors. So the receptor on the hepatic cells, here it is LDL or IDL receptors. These receptors on the hepatic cell recognizes, here in the IDL there will be triglyceride, then cholesterol esters, then ApoB100 and ApoE. So the receptors in this on the hepatic cell recognizes the ApoE and ApoB100 on the IDL. This receptors recognizes ApoE and ApoB100 on the IDL, and it is taken up by the receptor mediated endocytosis. It is taken inside by receptor mediated endocytosis, and here it is cholesterol ester, cholesterol, triacyl glycerol. Then by hepatic lipase enzyme, it is converted to free fatty acid and cholesterol. So majority of the IDL, these are converted to LDL. These are converted to LDL, that is low density lipoprotein. This is intermediate density lipoprotein. VLDL means very low density lipoprotein. LDL is low density lipoprotein. So majority of the LDL is converted to LDL. Function of LDL is to transport cholesterol from liver to peripheral tissues. Transport cholesterol from liver to peripheral tissues. So this IDL, most of the IDL particles are converted to LDL by losing ApoE and some triacyl glycerol. So LDL will become cholesterol ester, cholesterol, B100, that is LDL. So IDL, majority of IDL will be converted to LDL. This is by losing triacyl glycerol and ApoE, and it will become cholesterol ester, cholesterol and ApoB100. Then this LDL is taken up by, this is extrahepatic tissues, from mature VLDL by the enzyme lipoprotein lipase, free fatty acid and glycerol to the peripheral tissues. Then this LDL is taken up by the peripheral tissues. This is by receptor mediated endocytosis. Here also receptor mediated endocytosis. Then there is liver, and many extrahepatic tissues have these LDL receptors. These LDL receptors on the cell surface is in pits. These are in pits. Then these are coated by some of the proteins on the cytoplasmic side. That is known as Clathrin. This is known as Clathrin. So these pits are Clathrin coated pits. So the binding of the LDL to the receptors, this binding of LDL to the receptors is by ApoB100. So LDL receptors, or this LDL, it is taken up by peripheral tissues. This is by receptor mediated endocytosis. This LDL receptors on the cell surface is in pits. These are coated on the cytoplasmic side by some proteins. That these proteins are known as Clathrin. So Clathrin coated pits. So the binding of LDL to the receptors is by this ApoB100. So let's see the binding and internalization. So when the LDL, that is the ApoB100, binds to ApoB100 receptors, it will form a complex, that is the receptor LDL complex, and this complex is internalized by endocytosis. So let's see the mechanism. So when LDL, that is cholesterol esters, this is Clathrin coated pits. Clathrin coated pits. This is LDL receptor. LDL receptor. When this LDL, that is the ApoB100 part, binds to the B100 receptor, there will be the formation of a complex, that is LDL receptor LDL complex, that is internalized by endocytosis. So that time there is formation of endosome vesicles. So endosome vesicle means this cholesterol esters and this receptor, that is Clathrin coated. So it forms endosomes. Then it combines with the lysosome. It combines with lysosome. At that time, it combines with the lysosome. At that time, the Clathrin, there will be shedding of Clathrin. There will be shedding of Clathrin, and this receptor will be recycled. This receptor will be recycled and it returns to cell surface. So this is the receptor. It will be recycled and it returns to the cell surface. Then it will form lysosome. It will combine with the lysosome and it shed the Clathrin, and the receptor is recycled and it returns to cell surface. Then see cholesterol esters. Then after that, so inside the lysosome there are enzymes, that is acid hydrolases. Acid hydrolases enzymes, it hydrolyzes this cholesterol esters and the apoproteins in this particle. So the acid hydrolysis enzyme in the lysosome, this hydrolyzes this cholesterol esters and apoprotein into free cholesterol and amino acids. So this influx, then this cholesterol is translocated into the cell. This influx of cholesterol inhibits HMG-CoA reductase enzyme. This HMG-CoA reductase is the enzyme, that is the rate limiting enzyme in cholesterol synthesis. So the influx of cholesterol inhibits this enzyme HMG-CoA reductase and it suppresses cholesterol synthesis and uptake. So most of the LDL in circulation is removed by, that is about 2/3 of this LDL in circulation is removed by these receptors, LDL receptors. The remaining LDL is removed by scavenger receptors or by pinocytosis. So inside the cells, this LDL releases cholesterol. This cholesterol can either be stored or used. So inside the cell, LDL releases cholesterol. This cholesterol can either be stored as cholesterol esters, or it can be used for membrane synthesis, then bile acid synthesis, then for steroid synthesis. Then ACAT, acyl-CoA cholesterol acyl transferase. Acyl-CoA cholesterol acyl transferase enzyme promotes the esterification of cholesterol. This enzyme promotes esterification of cholesterol. So, the remaining portion of the LDL, most portion this will releases cholesterol. Then remaining portion of LDL, it is taken up by macrophages. It is taken up by macrophages. So, in the case of increased LDL, there is production of oxidized LDL. Oxidized LDL. Once it is internalized by the macrophages, it forms foam cells. Foam cells. Foam cells are fat-laden macrophages. These are foam cell means these are fat-laden macrophages containing LDL. The name foam cells, these are named because the lipoproteins give the cell a foamy appearance. Lipoproteins give the cell a foamy appearance. That's why the name foam cells. Then these foam cells can form a plaque. This can form a plaque that can lead to atherosclerosis. Atherosclerosis. So that is about metabolism of VLDL and LDL. This is today's topic. Thank you for watching.